think about Alzheimer's for a second. I mean, it's this truly devastating disease, right? It chips away at, well, everything that makes someone them, their memories, their personality, their independence. It affects millions, and we're all searching pretty desperately for answers, for solutions. But what if? What if we could spot it? Not when the memory loss starts, but years before, maybe even decades. Just picture that for a moment. What kind of shift would that be? The possibilities for, actually intervening, for prevention, for real hope. Okay, let's unpack this then. Today we're doing a deep dive into some really groundbreaking work. It's a study out of Florida International University, highlighted recently on SciTech Daily. And it's not just hinting at early detection. It seems they've identified a pretty remarkable new biomarker, one that could, well, fundamentally reshape how we fight Alzheimer's.
Exactly. And that's our mission here for you listening. We want to explore this key biomarker. It's called TSPO. We'll try to understand how it might just completely transform not only how we diagnose Alzheimer's, but maybe the entire approach to treatment strategies. What's really fascinating, I think, is this promise of getting involved, of intervening at such an incredibly early stage. It's like opening a window we just haven't had before.
You've really hit on the core problem there. One of the biggest challenges, the real frustration with Alzheimer's now, is timing.
Yeah.
By the time someone actually gets a diagnosis, when the memory problems are clear, undeniable, the disease has often been silently progressing for years, maybe decades.
Right. Significant damage has often already occurred.
Exactly. Irreversible damage in many cases. And that means any treatment we try now often feels like it's starting way too late. Its impact is just diminished. So the need for genuinely early detection, maybe even pre-symptomatic detection, is just critical. You really can't overstate it.
You really can't. And that's precisely where this protein, TSPO, comes in. TSPO stands for translocator protein 18 kilodea. Now, TSPO itself, it's not exactly new. It's a protein found mainly in the mitochondria of certain brain cells, particularly microglia.
Analytically, those are the brain's immune cells, right?
That's right, the resident immune cells.
Right.
And TSPO expression, it ramps up quickly when there's inflammation or injury. It's like an alarm bell for neuroinflammation. It's actually a well-established marker for brain inflammation. And the lead researcher here, Dr. Tomás Guilarte from FIU, he's been studying this specific protein for, get this, over 30 years.
30 years, wow.
Yeah. His work really laid the foundation, establishing TSPO as a reliable imaging marker for inflammation across, well, a whole range of conditions, neurological, neurodegenerative, even psychiatric disorders. It has a solid track record.
That long history you just mentioned, that's where this gets really interesting, I think. And it adds so much credibility, doesn't it? For you listening, this isn't some sudden flash in the pan. It's not an overnight discovery. This new research, it's really the culmination of decades of focused, dedicated work by Dr. Guillarte's team on this one protein.
Absolutely.
That kind of deep foundational science, it just gives these findings so much weight. It's built on solid ground.
It really is. And this current study, which was led by Dr. Guilarte, along with Daniel Martinez-Perez, he's the first author, a PhD candidate in the lab.
Yeah.
They took that solid foundation and built on it with a really innovative approach, actually.
So how did they do it? They didn't just sort of stumble on this.
No, not at all. It was very methodical. They used some pretty advanced imaging techniques. Think of it like a highly specialized brain scans. And they used these to track TSPO activity very carefully inside genetically engineered mouse models.
Okay, models designed to mimic Alzheimer's.
Exactly. Specifically, models of familial Alzheimer's, the type that runs in families and often starts much earlier. This let them watch the disease process unfold in a controlled way.
A sophisticated approach from the start, then.
Definitely. But, and this is really crucial, they didn't just stop with the mice. That's the validation step that makes this so compelling. They confirmed their findings using human brain tissue, invaluable donated tissue.
From actual Alzheimer's patients.
Yes, and not just any patients, from members of this unique community in Antioquia, Colombia. It's the world's largest group with early-onset familial Alzheimer's. They carry a specific genetic mutation called the paisa mutation. It was first identified by the late Dr. Francisco Lepra, who, incidentally, was also a co-author on this paper.
That's significant.
It is. People with this mutation often start showing symptoms in their 30s or 40s. Tragically, they usually pass away in their 50s. So if you connect this to the bigger picture, you have these cutting-edge animal models combined with insights from this unique human genetic group. It makes the findings incredibly robust, powerful combination.
That really does add a layer of certainty. Okay, so let's get to the core findings then. What did they actually discover, starting with those mice? What was the earliest sign?
Well, the first thing that really stood out was how incredibly early these elevated TSPO levels appeared and where they appeared. They detected the increase in a specific brain region called the subiculum.
Subiculum, that's part of the hippocampus, right? The
Like,
memory center.
exactly. Think of the hippocampus as the memory factory, and the subiculum is like a key shipping dock, crucial for sending memories out to be stored or used. Its early involvement is, well, concerning.
And the timing was the really startling part.
Astonishingly early, they saw these elevated TSPO levels in the mice as young as 6 weeks old.
6 weeks. What does that translate to in human years?
Roughly speaking, it's like seeing this Alzheimer's signal pop up in someone who's maybe 18 to 20 years old.
18. Wow. Long before anyone would even think about memory problems. That's an incredible early window.
It really is an unprecedented glimpse into the disease's very beginnings.
A potential sign of Alzheimer's showing up almost before you're fully an adult.
Yeah.
So where exactly was the TSPO highest? Within the subiculum.
They pinpointed the highest concentrations within the microglia, those immune cells we mentioned. And even more specifically, these activated microglia were clustered right around amyloid plaques.
Ah, the plaques. The classic hallmark of Alzheimer's.
Precisely, those protein clumps. So it strongly suggests a direct link. TSPO, the immune response via microglia, and the actual physical signs of the disease starting to form.
That paints a vivid picture. Inflammation right at the scene of the crime, so to speak. I also heard there was something important about differences between males and females.
Yes, that was another really significant observation. They found that the female mice consistently showed higher levels of TSPO compared to the males.
And that mirrors what we see in humans, doesn't it?
It does, strikingly so. It reflects that sad statistic that about 2/3 of Alzheimer's patients are women. So this finding in the mice, this sex difference in TSPO levels, it might offer a clue, maybe a biological reason, for that disparity in humans. It really highlights why we need to consider sex differences in this kind of research.
Absolutely. Could be a whole new angle for research, maybe even for treatment down the line. And what about how long this marker sticks around? Was it just an early blip?
No, and that's another key point. The human brain samples, the ones from the Colombian cohort, confirm the patterns seen in the mice. They showed that TSPO levels remain high in those microglia near the plaques, even in people who are in the late stages of Alzheimer's.
So it's persistent. It doesn't just fade away.
Exactly. It's not some fleeting signal. It seems to be consistently elevated throughout the disease course, which makes it a potentially reliable marker across different stages.
Okay, so let's recap. We've got this biomarker, TSPO. It shows up incredibly early, like late teens in human terms. It's located specifically in immune cells clustered around plaques. It's higher in females, matching human patterns, and it sticks around. So this raises a big question, doesn't it? What is this persistently high TSPO actually doing? Is it just a flag saying damage here, or is it actively involved, maybe making things worse? Or could it even be, I don't know, protective somehow?
That is the multi-million-dollar question right now, and it's exactly what the researchers are digging into next. Given how consistently it's there and its close link to the microglia and plaques, it seems unlikely it's just a passive bystander. Understanding its true role, is it harmful? Is it helpful? Is it maybe doing both, depending on the context? That's absolutely key.
That determines what you'd want to do about it therapeutically.
Precisely. Do we want to develop drugs to block TSPO? Or maybe drugs to enhance whatever potentially protective function it might have? Or something more nuanced? We need to know its function first.
So let's talk implications. What could this all mean for you, listening at home, for the future of dealing with Alzheimer's?
Well, it means the potential here is just immense. TSPO could genuinely revolutionize things, both diagnoses, obviously, but also treatment. It's not just about slightly earlier detection. This could be a total paradigm shift. Imagine doctors being able to use therapies, maybe neuroprotective drugs or anti-inflammatories, years, even decades before memory loss starts, essentially trying to head the disease off at the pass.
Preemptive treatment.
Exactly. This biomarker might be the key to designing real preventative Alzheimer's care, putting the brakes on before the damage gets out of control. The researchers themselves said, look, if this helps us delay Alzheimer's by even 5 years, that alone would dramatically improve lives and slash the overall prevalence of the disease worldwide.
A 5-year delay would be monumental, absolutely life-changing for millions. And the team is pushing forward. Their next crucial step involves working with a new mouse model. They've developed one that completely lacks the TSPO protein.
Ah, so they can see what happens without it. Exactly. By comparing these TSPO knockout mice with normal Alzheimer's model mice, they can really pin down its function. Does removing TSPO make the disease better or worse? Does it protect the brain or harm it? Answering that will tell them whether blocking TSPO or maybe even boosting it could be a viable way to stop the disease progressing.
Makes sense.
And crucially, they're also expanding beyond that specific familial early onset type. They're now including samples and data from sporadic late onset Alzheimer's cases.
Which is the vast majority of cases, right?
Over 90%. So this is vital work to make sure these findings aren't just relevant to that rare genetic form, but could potentially help the huge number of people affected by the more common late onset Alzheimer's. It's about ensuring broad applicability, connecting the deep dive to real-world impact.
It really brings it home. And it highlights the personal relevance, doesn't it? For anyone who might be touched by this disease someday, directly or indirectly. Daniel Martinez Perez, the study's first author, put it really well. He said, "The more we understand these processes, the closer we get to tailoring treatments that can truly help before it's too late."
A powerful sentiment.
It perfectly captures the hope and the urgency behind all this research. So there you have it. Our deep dive today uncovered TSPO. It looks like an incredibly early, persistent sign of brain inflammation in Alzheimer's. It potentially gives us this unprecedented window, decades before symptoms, to see the disease starting. It could be a genuine game changer for how we understand, prevent, and maybe one day treat Alzheimer's.
It really could. And this, leads to a fascinating thought, something for you to really ponder. If diagnostics like this, based on TSPO or similar markers, become reality, if we can reliably identify Alzheimer's risk when someone is, say, 18, 20, 25 years old, how might knowing that, having that biological glimpse into your potential future, how might that influence the choices you make in life?
Big ethical questions there, too.
Huge ones. What does it mean for medical ethics? How does society adapt its whole approach to health, to prevention, not just for Alzheimer's, but for other neurodegenerative diseases? What new opportunities, but also what new responsibilities arise when we gain the power to see so far into our own biological destiny?
Really powerful questions to think about. Definitely something to consider as we watch this science unfold. We hope you'll continue exploring these ideas. Thanks for joining us on this deep dive.